Thank you very much for all of you to take your time and join me in this session. This is the BioEdge series, lecture number 11. For consuming bandwidth, I'm going to close my camera. Okay, so this is the 11th lecture, what we are doing in the series called as BioEdge series, and I think only one more is left to be completed. And we would be more than happy to host you in the next session, which is going to be the last session of the 12 lecture series we started under the banner of BioEdge series. We have been discussing different things in the technical side of the series, like discovery pathways, and we spoke about the opportunities, Indian opportunities, and so many things. Artificial intelligence we spoke about. So, today I would like to spend some time in the area of understanding how to actually approach the research job opportunities, because this is a very different space. So in this space, it is very important for us to understand how to actually approach the research jobs. Because sometimes what happens is we normally keep it this way in our mind. When you are in a college, when the student is in a college, they really look up for ROI. What is the return of investment they have made? What is the fees they have paid? What is the all the money put together? What is the return I am getting? That is one way of looking at it. When it comes to the job, I always encourage the candidates to look into IOR. When it's IOR, it means that it is the industry role, industry organization and role. So industry, organization, and role. When I say industry, today we are going to focus completely on pharma and biopharma and contract research organizations. And when it comes to role, we are going to talk about drug discovery and allied roles in R&D. How to approach that. Okay. And of course, when it comes to the type of landscape India is possessing, it is really very, very high. We have a large skill and employment landscape in India, particularly in the areas of medicinal drug. Medicinal drugs, when we are talking about, there are two types of drugs we are talking about. One type of drug which is already in the market, which is getting repurposed for something else. Or we are talking about a completely a new drug that is being discovered. These are the two things we are talking about. Please don't get confused with the term drug discovery with the biosimilar, because in biosimilar also we are going to use the same term drug discovery, though it is not a discovered drug, though it is not a complete drug you are discovering. You are developing something which is very closely related, both structurally and physiologically, to the patented molecule which is already existing. So I hope all the five of you in this session would be familiar with the difference between generics and a biosimilar. For a general understanding, I will repeat it. When you develop a small molecule as a similar therapeutic, keeping the patented molecule as a reference drug, then what you are developing is called as a generic drug. If you are developing a large molecule, for example, a protein or complex protein or a peptide, whatever you are developing, this molecule, if you are developing against or keeping a large molecule as a reference, then you call it as a biosimilar. That's the understanding. So in this particular session, we are going to talk about both. We are talking about both type of drug discovery landscape. One quick disclaimer is that this session is not going to cover the drug discovery pathway, which we have already done. We have already done the drug discovery pathway. So mainly I'm going to focus on how to understand and how to approach the opportunities that are available in the employment, in the drug discovery landscape. So what mainly we are going to see is to start with the big picture. When I say big picture, I'm talking about the country level approach. Give me one second. I'm talking about a country level approach. When I say country level approach, every country has its own domain specificity. For example, you have a kind of population. For example, let's say US population. US has a given population. In this US population, there is a specific type of disease which is major, which is contributing to the major segment. And then there is next one or the next one. So basically, how the country will approach this market is that how much money I have to spend in understanding how much or how many molecules that will be developed or discovered to treat the diseases in that particular cluster. If you want me to take an example, let's say India. In India, if you ask me what is the largest disease segment, mainly there are two disease segments. One is diabetes, cardiovascular diseases, and cancers, different types of cancers. So these are different segments. These are not the only segments. I'm just taking example of large ones. If you take these three things and then see how government is going to look at funding these type of funding discovery of molecules for these type of indications, these type of diseases. These are all disease clusters. These are all not diseases. For example, if you take an example of diabetes, diabetes is just a situation. It is a condition. This diabetes can lead to cardiovascular disease. Diabetes can lead to skin disease. Diabetes can lead to a muscular dystrophy. Diabetes can lead to neuropathy, diabetic neuropathy or nephropathy. Then it is a kidney dysfunction. So these are all disease clusters. So these disease clusters, when it is looked at from a country level, it is looking like this. The reason why we need to understand this particular approach is because each country is going to allocate certain amount of money in each of these segments so that these segments can be supported, well supported. Research is well supported. I'm saying the research that is happening in the research centers, government research centers, and so on, to some extent to the private research also. In the graph in the left-hand side, I'll take a pointer here so that can use it here. Yeah. So if you see the left-hand side, these are the stages, and these are the number of molecules, and the molecule numbers is categorized based on the size. When I say size, this not the size of the molecule, it is the size of the value. How many $1 billion worth molecule it is? That is the total size. So for example, let's say when it comes to the mega sized molecule. When I say mega sized molecule, again I repeat, we are not talking about the molecular weight. We are talking about the valuation of this drug, which is more than $200 billion. Then what is the disease cluster we are talking about? That is how this numbers to be understood. And when it comes to the left hand side, we are talking about the actual pathway. These are the stages. The first stage is the discovery. The discovery includes the target identification, the hit identification, the target lead interaction, then the lead optimization, then the target, then the candidate identification and preclinical development. These are the discovery. After the preclinical development is over, you get to do the preclinical trial. That is the animal trial. After the animal trial is successful, then you will be filing something called as the IND. That is investigational new drug application. The investigational new drug application is filed, and then if the IND is approved, then you will be allowed to do a phase trial. Phase trials 1, 2, 3 are the clinical trials that is conducted on human being. And then it goes into the registration. That is a complete pre-registration and registration process is the regulatory work where the regulatory department is mainly working on two documents to get two documents, a manufacturing license and a marketing authorization. So this is the overview of how a country level picture has to be understood from the number of molecules that are getting developed in each disease cluster. That is going to decide what type of talent the country needs. For example, today you are doing a PhD, or tomorrow you are planning to do a PhD. This information is going to give you a clear idea. If I select one particular cancer today for my research area, and if I complete my PhD and postdoc, will I be employable? After 5 years, after 6 years, will I be employable? This question will be answered if you understand what are the major disease clusters to which the country is supporting. The country is investing in, supporting in, and so on and so forth from a government perspective. Also, you have to do a little bit research on what is the segmentation from industry perspective. That is my next slide. From industry perspective, if you see, it is, I'm just taking 4 different examples, so don't worry about the business of the slide. Each organization will have an organizational level drug discovery pathway or pipeline. We call it as a molecular pipeline. When I say molecular pipeline, it is something like this. For example, let's say India is heavily investing on one particular technology. This technology will gradually mature. When this technology matures, India cannot stop investing on this other technologies that are emerging. Okay. For example, let's say mobile phone. India is investing on one particular type of mobile phone. We invested in 2G, or we used in a telephone, the regular telephone. Then we moved into 2G. After 2G, the landline got extincted, or it is getting extincted. Now, after the 5G mobile, 3G, 4G mobile, and 5G mobile comes, the 2G mobiles are technology is getting extincted, which means that when these technologies gradually getting mature, you have to keep investing on it. Then only the segment will grow. When it comes to the industry also, we call it as the molecular pipeline because the molecule in which you are investing will also mature. For example, today I am investing on metformin. Metformin is an excellent molecule for type 2 diabetes. I'm investing on it. When I'm investing, this metformin will be discovered, will be developed, will go through the preclinical trial, will go through the clinical trial. By the time the metformin is going to the clinical trial, if I'm not investing on the next molecule, could it be insulin or could be a biosimilar like glargine or any other molecule which is close to metformin. If I'm not investing on it, what is the biggest risk I'm taking as a company? The biggest risk I am taking is when metformin reaches one particular level, and for whatever reason, if metformin fails in the trial, then I don't have anything to move forward. That cannot be the case when it comes to a commercial organization or a private organization, a drug manufacturer. A drug manufacturer cannot depend on only one molecule. He can always depend on the organization can always depend on one disease cluster. In that disease cluster, we have to have multiple options available with us, and it should be gradually added in the downstream, gradually invest in it and move it upstream. Which means you're going to invest in the discovery and then invest in preclinical trial and development, move it to clinical trial. By the time the second molecule goes to the clinical trial, you have to identify the third molecule. That is how the R&D department is busy always. They have one project or the other to keep working on, which also means as a student, we need to be extremely diversified in our knowledge. Because for today's moment, we do not know which company we are going to join. So it's always a good idea in a postgraduate level to be little open. When I say little open, your projects should be little more technology oriented, not molecule oriented. It can be oriented to a disease cluster. It can be oriented to oncology. It could be oriented to diabetology or cardiovascular diseases and so on and so forth, or anti-infectives or so on and so forth. It can orient to one particular disease cluster. but certainly not one particular disease or one particular molecule. It can be an example. In that way, what happens is your opportunity to work in any other molecule is very high, which means that in your project, you are proving that I know how to approach, I know how to deliver. That's all you have to create. That's all you have to create as a part of your project, whether it is your MSc project or MPhil project. When it comes to PhD, it is slightly different. When it comes to PhD, what is very important is the orientation. Now you have to be oriented to that particular disease. In that disease, which particular biomarker you are going to work on. For that biomarker, then you have different areas. Like, are you going to work on a small molecule, large molecule, natural therapeutics, or whatever options you have. So when you design your MSc project or MPhil project or your PhD problem statement, that is a research area, it is very important for you to understand in my country what is the prevalence of diseases, which disease is more prevalent, which means that there is a possibility that the government is going to invest on it and do a research to find out is there is a alignment between the investment made by the government and research done by the private organizations are aligned to that particular disease cluster, to that particular indication. This will give you a clear idea of why particularly you have to look into the R&D as your career aspiration. So it is not easy to really do a decision or make a decision, particularly when it comes to R&D as your career. So how to go about now? Now, there are two things we spoke about so far. One is the country level approach to the molecules and the disease clusters. Second is the organizational level where they will keep adding up the molecules into the pipeline. Actually, these two are interconnected. The number of molecules they add up to this pipeline contributes to the overall approach of the country to that particular disease areas. Up to here, we have seen. Now, what is the way forward? Way forward, number one. When it comes to the research area, one of the research area I am taking DMPK, because DMPK forms the core of your drug discovery. Not only discovery, it also plays a very important role in the development, drug development. So it plays a very important role in drug discovery, also plays a very important role in drug development. That is the reason why if you see, majority of the time, the molecule's success is dependent on how clearly we are able to understand the adsorption, distribution, metabolism, and excretion of a drug. Because these drugs behave very differently between in the lab and in the body. For the reason, in the lab, it will be a very successful molecule. But when it comes to the real life situation, we don't have a complete idea over what are all the pre-existing conditions the patient is having and what are all the medications they are taking. So there will be a possibility of drug-drug interaction. When it is a drug-drug interaction, we don't have a clarity on how this drug is going to be successful. That is where the DMPK plays an important role. The drug metabolism and pharmacokinetics plays a role in understanding drug-drug interaction, drug-protein interaction, drug-food interaction. So if you understand all this, then there is a possibility that when you're developing and moving forward with the drug, you are moving forward with a complete visibility of how this ADME profile is going to look like for this given drug along with the other drugs, because you have a visibility on DDI. As I was mentioning to you, the importance of DMPK, when I say DMPK, I'm talking about the department and those who are working in the DMPK. I'm coming to the skill set in a while. Those who are working in the department, they are the one who are responsible for taking a crucial decision of go, no go. This is a very, very important decision organization takes when it comes to a molecule, which means should I move forward or should I stop? Because it's not only about technical moving forward. We are talking about a commercial moving forward, investment. Huge amount of resources has to be deployed to support the movement of this molecule. This gives us another very important clue that how we are going to utilize the in vitro, in vivo data using a in silico data to predict the pharmacokinetics. This is very important because on one side you have huge amount of data, on another side you have to take a decision whether it is go or no go. So now you have a combination of evidence-based discovery and structure-based discovery. This data can be run in silico to understand whether I can take a decision to go or to no go. What are the very specific areas in DMPK? This gives this type of decision points are number one, the in vitro assay. As all of you can imagine, the first one is in silico modeling. Once the in silico modeling is completed, when I say modeling, we are talking about structure elucidation, 2D structure elucidation, 3D structure elucidation, quantitative structure activity relationship, QSAR, and all that. So basically, once you develop your in silico model, then you have to validate it with in vitro models. This in vitro model is going to give a lot of information later for you to use the artificial intelligence to analyze this data. Artificial intelligence can provide meaningful information only when it has a meaningful data. This is a primary understanding, which means that the meaningful data generation comes from the R&D department. It could be a cytoplasmic protein inhibition or microsomal stability, whatever in vitro studies you are doing. This data that is coming out is going to be very important. Whenever a drug is designed and delivered, what is the next step? Is to take sample from the patient to analyze in the laboratory to determine the ADME profile. That is where the bioanalytical skills comes into picture. Whether it is liquid chromatography, mass spectroscopy combination, or it is mass spectroscopy, what is the most important skill you need to have? Number one, you need to know how to use it. For that, you need to know how it works. If you know how it works, and if you know how to use the liquid chromatography mass spectroscopy, the next level you need to know is, if you are using that, will it provide the dependable value, dependable data? For that, you need to do method development validation. These are the most important areas when it comes to bioanalytical skills, because bioanalytical skills are very quantitative oriented. It is not quality oriented. It is not qualitative oriented, not quality. It is not qualitative oriented. It is quantitative oriented. It's all numbers. So bioanalytical skills is very, very important. Next to in vitro assays, bioanalytical skills makes the firm foundation for you to be successful in R&D space. There are certain PK modeling and PD modeling. When I say PK, I'm talking about pharmacokinetics and pharmacodynamic modeling, where one model will lead to the another. So far we have talked about is the non-compartmental analysis. But when it is a connection between PK and PD, then it becomes a complete non-compartmental analysis. I don't have to tell you what is the importance of data science and statistics when it comes to decision making. Huge amount of data will be generated. Now, the first thing is, before even you start analyzing the data, will you have, or do you have the capability? Do you have the flexibility? Do you have tools to clean up the data in first place? That is where Python comes into picture. Python comes handy in cleaning up the overall data which is going to be used and analyzed. Then you have algorithms developed and algorithm scaling up and all that. And of course, statistical model. When I say statistical model, it could be a design of experiment, or it could be SAS, or it could be SPSS models, and so on and so forth. So basically, you are going to focus both on data sciences and statistics simultaneously to make an informed decision. Once the decision is made, then it is very important to go for a prediction. This is where I was telling you, you cannot depend only on physical data, not only on evidence data. You have to have a combination of physically generated data along with evidence-based evidences, data evidences, and then run it through machine learning. Then it gives you a clear prediction, molecular descriptors and QSAR models. Then you try to understand, is it really worth to go, or it's an early decision we can take to say no go. What is the biggest advantage in this approach of a combination of the in silico and in vitro data or data-based, evidence-based discovery decision is that you won't face any problem when it comes to submission, the regulatory work, whether it is FDA or European Medicinal Agency for filing of investigational new drug, or it is a new drug application submission. This data package is going to decide your clinical strategy. So in Crux, there was a period when uh, more importance or more significance was attached to the in vitro work. Then there was a period when bioinformatics came in. There was a lot of work that was done in vitro has been were taken over by the bioinformatics. Now we are in a situation where we have the ability to check best of both sides. You have data from in silico, you have data from in vitro, put together and ask the machine to learn that and give predictions. Based on that, if you are going to conduct your clinical trial, the possibility of failure is extremely less. How AI is transforming drug discovery? I have made one lecture on this itself separately, but to shorten to put it in a nutshell, whether it is a small molecule or it is a large molecule, whether it is a small molecule or it is a large molecule, AI tools are available for for you to use. For example, let's say 1 minute. Yeah. So, uh, as good as bioinformatics, we have a very strong data set on chemoinformatics. So, whether it is a small molecule or it is a large molecule, we have a lot of data that is available. Therefore, it is easy to do little bit of stronger algorithms like GAN. The GANs are the generative adversarial neural network. These GANs are transformers models, which is a LLM and SLM based models. This gives a lot of information which actually replaces the work of medicinal chemist, because iteration by the medicinal chemist is a very huge data that is putting up the final picture. That iteration is a very important one. They spend weeks and weeks time to actually get a correct iteration, but now it is very handy to make use of the AML model with chemistry database to do the iteration very simple. Then it comes to ADME prediction. Now we have added one T to the ADME profile, that is the toxicity profile. Because as you can understand, all the medicines will have some impurity or the other. Only thing is it will be always available in the lesser quantity than that is prescribed by the regulatory agency. So these impurities are going to give one or the other side effects. So it is very important for us to understand the entire toxicity profile of the molecule. So I give examples of the cytoplasmic protein interaction studies, using your the regular ones, the SMILES ones, that is your simplified molecular input line entry systems. So these systems will give excellent prediction on your pathway level outcome on ADME. I mean ADME-T, admit. Okay. And then target identification. Target identification is extremely NIP, NLP based work, because it is more to do with 2D elucidation, 3D structure elucidation, and so on. So NIP, NLP makes a very important multiomics platform to gain the identity of a novel drug molecules, or particularly druggable targets. So this heavily depends on the bioinformatics. Structure-based data is very important to create the basic data that has to be later validated by in silico. So the computational biology finds it very important. Data analysis and data science makes a very important dent in the clinical trial AI. The clinical trial artificial intelligence is mainly based on how efficiently you are going to stratify your patients. How are you going to predict this particular patient is going to drop out from the trial based on the trend? Because you cannot exactly say this person is going to drop out, but at least you can say this many percentage of people are going to drop out, and these people belong to this particular mindset. So that type of prediction can come in. In those type of real life evidences, which when you do the mining, it will accelerate the phase 1, phase 2, phase 3 clinical trials, because you already are informed that this many patients will drop out from your study. So you already have a plan. You can already have a plan to replace with more volunteers at the time of phase 1, phase 2, phase 3 clinical trials. My favorite is process and formulation, because I have seen so much information, so much modification, so much enhancement that has happened because of machine learning for formulation development in the generics market. Because in formulation development, I'll take only one example for DOE and ML. How to mix the API with the excipient and solvent. Very simple problem statement. But we used to take so much time, so much resources spent to come to a conclusion which solvent to be used, what are the excipient that goes well with that solvent and do not create much of reaction with the API. This comes very handy with a very simple agent finder, which is a process and formulation AI. So the process chemistry goes through a very important transformation, because when it comes to India, India is hosting 200 plus AI pharma projects. There's large organizations like Syngene, Biocon, Dr. Reddy's, Cipla, Lupin, Davis Labs, Piramal, and so on and so forth. I'm not talking about only the large organization. In addition to the large organization, we have a very strong startup ecosystem. Okay, this startup ecosystem is also working very heavily on use of AI in drug discovery and development. In addition to the big and small, I mean, medium scale contract research organizations, for example, like IQVIA and so on, and the PPDs and so on. So, what is important, or what is the take-home message from this particular conversation is this particular situation, this particular transformation is creating lot of roles, which are very new roles, new age roles. These were roles which were not existing in the R&D space. So it creates roles for both life science students and computer science students. But the catch here is, it is not only for the computer science students, or it is not only for the life science students. It is a combination. It is a combination of life science and computer science, or it's a combination of computer science and life science. Either ways, wherever you are in. If you see the sectors which are working on it, contract research organizations, it could be Syngene or Anthem or Jubilant, Lambda, Eurofins, and so on and so forth, where the main the main resource requirement is DMPK scientist, bioanalytical scientist, the pharmacokinetics modelers. Because earlier we used to have a very important role in IT segment. I'm not getting the name. Coders, encoders. So like that, very important role has come in the R&D space, which are the PK modelers. Then you have biopharma. It could be, Biocon, or Serum Institute, Biological Evans, or Biological E, Bharat Biotech, and so on. I'm just taking few examples. So if I'm not touching upon any particular company, that doesn't mean that it is important, not important, or it is not working on it. I have just taken few examples, that's all. In generic space, if you see, Sun Pharma playing a very important role, Cipla is playing a very important role, Lupin, Dr. Reddy's, Arbindo plays a very, very important role because they are very cautiously, they are very consciously investing in development of these areas as a specialized areas for ADMET modelers, process artificial intelligence scientists, and particularly in regulatory sciences. There is a huge scope for using AI in regulatory sciences. Coming to innovative pharma molecules, like those who are heavily working on NDAs, it could be Glenmark, Alchem, Zydus, Piramal Days, Biocon, etc. Tech pharmas, tech pharmas like TCS, for example. When I say TCS, they are into the services, pharma and biopharma services. They are doing clinical data management services. Plays a very important role for including machine learning engineers for drug related data. As I told you, as good as bioinformatics, now we have a strong chemoinformatics, and we have natural language processing specialists who can develop algorithms which can only listen to the patient conversation and convert it to a narrative. And regulators, very, very important. The global regulatory affairs teams and clinical regulatory affairs teams. So these are the areas where the key employers, as you can see in the second column, are investing in. Your skills should be aligning to them in the beginning, and gradually moving forward, the skill has to improve. So as you can see in the right-hand side, as a fresher, you may join as a research associate or a junior DMPK scientist. But over a period of time, you still have a long career path in these domains. The reason why I wanted to specifically tell you this particular segment is because sometimes we may get a stigma or we get a fear that it is a new area. If I set up my career in this area, will I have a career growth? This is your career growth for your new age type of jobs. Freshers, associate, senior, principal, principal scientists or group leaders or chief scientific officers and so on, and director, DMPK, VP Discovery, and chief technology officers and so on. So you have a very strong career path also. Only one thing I would like to emphasize and leave the thought with you is that whatever time you are investing, whatever money you are investing in this particular area, in this particular domain, whether it is computational chemistry, DMPK, PK modeling, artificial intelligence and life science and drug discovery, please remember you are not spending. You are investing in country's highest growth career pathways, which means that if you are worried that today if I invest a couple of lakhs in this, is it going to give me directly this job? If you are lucky, yes, maybe, may not be. But the investment what you have made is going to be a career-long benefit investment. Because this knowledge is going to propel your career growth in whichever department you are joining in. It could be DMPK. Please remember, in India, you have so many colleges and institutions who are churning out pharmacists and chemists and biochemists. Still, it is underserved. DMPK is underserved. The reason why I'm saying it is underserved is not from the number of manpower perspective. number of trained manpower perspective, skilled manpower perspective, that portion is underserved. When it comes to AI in the other areas like ADME profiles and toxicity profiles, the AI skill is going to really multiply your value in your organization. And please remember, India is going to be your launch pad for propelling your career growth from India to outside also. Because if you are going to have a spectrum of experience in India in all these areas, it is going to be extremely easy for you to get into the global market and hit the job market in the global level. With this thought, I'm going to leave you. If you have any questions, you can let me know. Thank you very much. Are there anybody who is continuously attending these BioEdge series? I know Dr. Kavisha, for sure she's attending this continuously without missing any session. Are there anybody else who is continuously? If you can raise your hands, I can see that. Aditya. Oh, wow. Excellent. Thank you so much. I just want to hear from you how useful this series is. Aditya and Kavisha. No, I mean, Dr.
I'll just
Kavisha.
unmute now.
Yeah. Now, do they have a capability to unmute?
Good evening, sir. This is Dr.
Good evening,
Kavisha here.
doctor. Good evening, doctor.
How are you doing, sir?
Absolutely good. Thanks for asking. How are you?
I'm fine, sir. Actually, sir, your webinar series is so structured that we can get a glimpse of understanding that what is the recent status and what is required, what skills we also as a teacher also we should understand and be able to guide our students. So that's one of the biggest plus point for me as a teacher. When students ask something, we also need to update ourselves to understand
Yes.
what
Yes.
they need to study, or we need to guide them so that they can get those skills to get into the industry. So that has been the plus point for me.
Thank you so much, doctor. Thank you very much. Yes, Jester.
Good evening, sir.
Good evening.
Sir, myself Aditya. I'm a
Okay.
BTech student,
Okay.
second year. I'm working on bioinformatics and all with my career. So the thing is, I am getting knowledge from people like you and every attending webinars and all. From Biocon itself, I'm getting a lot of things. Uh, like how to work on it and how is the perspective and the future of bioinformatics and all.
Very good. Excellent.
I'm currently working on admit and R programming and all.
Oh, very good.
Yeah.
Very good.
Thank you so much,
Nice to
sir,
know.
for giving us such a good opportunity to attend those
Anytime, anytime. You're
sessions.
most welcome.
Thank you, sir.
No problem.
So actually, what I wanted to ask is, like R&D work is just an project based, but it will be not involved in a production at all. So if we select that, how come our it's only if we have the skills of equipment, is it enough for itself, or we need to have industrial visit and industrial hands-on work? We have to.
Okay. What I gather from what you have asked is there are two things in this question. One is if I'm specializing in R&D, Will you have a capability to go into production also? Number one. Number two, to be in industry, is your skill alone is important, or you need a training in the industry itself?
Yes,
Is that what I'm gathering?
yes, sir. That's important question.
Okay, so I'll go one by one. The first one is, as far as industry is concerned, we call it as fungibility. Fungibility means you join in one department and then you become fungible to other departments. I'll take example of the same two things you have told me about R&D and production. You join in R&D, you work in R&D for a year or two, 3 years, 4 years, you work. When you're working, if you're working in upstream process development, then from upstream process development, you can go into a department called as MSAT, Manufacturing Science and Technology, which is a bridge between production and R&D. From MSAT, you can enter into production. This is one line of going. So it is not that you are, if you are joining in one department, you are going to retire in the department. It doesn't happen that way. Always you can move internally into other departments as long as you are equipping yourself and working in correct departments. This is the first portion. The second portion is, even today, the freshers are getting job without any problem, uh, but the number is less. The number is very less. What is the difference between those freshers who are getting job and those who are not? Is that the application know-how. Those when you are studying, for example, when you study chromatography, you know that chromatography is a purification method, purification technology. Now, if you try to understand what is the basic principle of chromatography, it is a partition coefficient. Now, keeping this principle in mind, if you are going to try to understand in what are all technologies, partition coefficient is a basic principle. This is how you are getting prepared for the industry job. If you are going to focus and restrict yourself only to the syllabus, then you need little more training and you need little more upskilling and skill development, and then you have to get into the industry. What Biocon Academy is doing is just the connection between your academic deliverable and industry's expectation. That's exactly what we are doing. I think you're saying bye. Thank
Yes,
you,
sir.
Justin.